Well, good afternoon, everybody. My name's Carl Larson from RMCG. Hope you're well. Today, we're covering in control managing capsicum viruses for profitable vegetable production. And we're really privileged to be joined by Denis Persley from the Queensland Department of Agriculture and Fisheries, who is based up in Brisbane and will be talking to us today about some of the key findings from the area-wide management of insect-vectored viral and bacteria diseases project that was recently completed. This webinar is being hosted by the Soil Wealth and Integrated Crop Protection Project today, which is a collaboration between RMCG and AHR and funded by Sport Innovation using the Vegetable R&D Levy and funds from the Australian Government. It's now in its 8th year of delivery, and the main purpose of the project is to get R&D out on farms and in the hands of growers and agronomists, which brings us to events like today, the webinar series. So this is the second of 3 in the area-wide management mini-series. If you did miss our August session with Craig Webster from the Department of Primary Industries, Western Australia, you can catch up on that and all things curcumin viruses. And we've also got a session in October with Cliff Canotti from Agriculture Victoria that will cover specific viruses related to lettuce. If this is the first webinar that you've joined, the go-to control panel is on your screen. There are a couple of things I just wanted to highlight quickly. One was the handouts. So there's a couple of relevant resources for you to click and download and take away from today's session. And importantly, a chance for you to interact with Denis and ask those burning questions. So at any time throughout the presentation, please feel free to type those in and they'll come through to me and we can put those to Denis at the end of the talk. We've just got half an hour today, so pretty short and sharp. So without further ado, Denis, welcome and over to you.
Thanks very much, Carl. My talk today is on virus diseases in capsicum, but I'm also taking the opportunity to talk a little as we go through about the ways or the main means which viruses are actually spread from plant to plant. I think it's important to realise that in many cases this is a very specific evolved process rather than just a particular insect having to get contamination as it moves through a crop. So we'll move through the slides, and the first group I'd like to talk about are viruses spread by thrips, and that's particularly the orthotosmoviruses, as they're called, tomato spotted wilt, and capsicum chlorosis virus. This group of viruses, about 30 species worldwide. They're all spread by thrips, and tomato spotted wilt virus is certainly the most widespread and probably the most damaging of viruses in capsicum and chilli in Australia. It occurs in all states in both field and protected cropping. Interestingly, there's probably several thousand thrip species that can feed on plants, but only about 20 of those thrip species are vectors or carriers of the tosmoviruses. And the spread is a result of a very complex cycle between the thrip, the virus, and the host. And it's probably the most complex of the virus transmission cycles for plant viruses. It's persistent transmission, which I'll explain, and the virus also multiplies in the thrip insect as it goes through that particular species. So just example of some symptoms of tosmoviruses. We have mottling and mosaic and spotting on the leaves of the plant. This is on the left. We have a virus called capsicum chlorosis virus, and down here is tomato spotted wilt virus, and both can cause really distorted, disfigured fruit, which clearly, is unsaleable. And this is where some of the really major losses come, because if you have a lot of infected plants, particularly infected early, then almost all the fruit from those infected plants will be unsaleable. So you can have really major losses because all that production's gone. So tomato spotted wilt has a very wide host range. In fact, it infects over 600 plant hosts. In Australia, the main hosts are capsicum, chilli, and lettuce and tomato. The major vector or carrier is tomato, is the western flower thrips and tomato thrips. And I guess the real issue internationally with tomato spotted wilt really came once the western flower thrips started to move internationally. With that followed huge outbreaks of tomato spotted wilt virus. Has many weed hosts, including sow thistle, in North Queensland snakeweed, which is the weed over on my right, and then in southern areas, you know, things like capeweed can be a really major host of both the virus and the thrip. Capsicum chlorosis virus is in a separate group of tospoviruses. It's in the watermelon silver mottle or serogroup 4 group, which are very, very widespread throughout Southeast Asia. The main vector in Southeast Asia is thrips palmi, whereas in Queensland, it's probably the tomato thrip. And interestingly, western flower thrip isn't a vector of capsicum chlorosis virus, which goes to show the specificity that can sometimes occur with the insect vectors. This virus has a really wide and varied host from capsicum, chili and tomato through to orchids and pineapple. So it doesn't discriminate very much in terms of which plant family it likes to go into. It's been particularly important in North Queensland in the last year or so. This season we've had quite high incidences of CaCV in the Eyre Gumlu area, and it also occurs at Bundaberg. The distribution of the virus, particularly at Bundaberg, is really related to an important weed host, blue billygoat weed or ageratum, which hosts both the thrips vector and the virus, and there's often a good relationship to how much weed you have on the headlands and in wasteland and how much capsicum chlorosis virus you're actually getting in the crop. So, you know, 2 distinct viruses, tomato spotted wilt, largely, or more importantly, maybe in southern Australia, and then as you go further north into the tropics, capsicum chlorosis takes over as being usually the dominant virus. Now, if we look at the life cycle here, can we just move the photographs to the right a bit or make them smaller?
We're in slideshow view, Dennis, so
Okay.
the image is static for this particular part.
Okay, that's okay. So just with the life cycle of the thrips, we actually have it's a complex one that the adults lay the female eggs on the plant and the larvae feed, and it's those first and second stage larvae which actually pick up the virus from an infected plant, and then the virus actually has to travel through the gut and the internal organs of the thrip, and as it does so it multiplies and then goes through the pupal stages which don't feed, but the virus continues to multiply within those stages, and then the pupa emerges as an adult, and it's the adult thrip which is active, of course, and by that stage it has a full complement of virus particles, and that's the one that moves through a crop and infects them with the virus. Now, that thrip can actually carry virus for the remainder of its life. It probably needs to feed for maybe 15 or 20 minutes on a plant to actually infect it, but it retains the virus in the insect for the rest of its life. And the other interesting thing is if the adult feeds on an infected plant, which it will, if it's not already full of virus, it may absorb virus, but it can't actually transmit. So for a thrip to be able to transmit tospovirus, it has to actually get it as a young larval stage, have it multiply through the insect as it grows, and then the adult is able to transmit it to new hosts. So it's a very complex transmission cycle and indication that in many cases only one type of insect can transmit one type of virus. They're quite specific in their requirements for transmission. So the next stage is viruses spread by aphids. Aphids are the vectors of the largest group of plant viruses called potyviruses. Potyviruses are important in capsicum worldwide, and we're extremely lucky in that many of those potyviruses that infect capsicum aren't present in Australia. The one that we do have here is potato virus Y, or PVY, and again that causes some sporadic losses in capsicum in different districts, but most varieties are now resistant, so the incidence or level of PVY in capsicum has decreased a lot, I'd say, in the last decade or so. It's not as good in chili, where many chili varieties still seem to be susceptible to PVY. Now, cucumber mosaic virus is a member of another virus group, but it's also spread by aphids. And again, it's an important virus worldwide. And in Australia, we can see it particularly at Carnarvon in WA, when a number of seasons become really important. And as I mentioned before, we're still free of many of the potyviruses that cause a lot of damage to peppers and capsicum throughout much of the rest of the world. So on the right of the screen here, there's potato virus Y infection, and it particularly causes green vein banding along those main veins. And then on the left, we've actually got a capsicum that's got cucumber mosaic virus symptoms. You know, a lot of mottling, stunting, mottling patterns on the leaves. So now I'd like to mention the difference between non-persistent and persistent transmission. I think these are two important concepts. Like a non-persistently transmitted virus, as the name probably suggests, it happens really quickly. That an aphid or another insect can actually pick up a charge of virus from a plant in less than 30 seconds and also then transmit it or spread it to another plant in that same period of time. So all that happens is that the aphid, with this little needle-like stylet, puts the stylet into the epidermis, into the young tissue. A charge of virus sticks on the stylet, and as it moves, it can then inject it into another plant. And the aphid can only retain that virus on its stylet for a matter of hours. If it doesn't infect another plant during that period, it loses it and needs to get a recharge of virus for another attempt. So a very rapid process, no circulation through the insect, just on the stylet or the sucking mouth part. A persistent virus, on the other hand, and this applies to tomato spotted wilt that I mentioned before, and to another group I'll mention in a minute called bigamoviruses. Persistent viruses, the insect requires feeding for a number of hours to actually get a charge of virus. The virus then has to circulate through the body system before it can transmit again. So it's taken up by the stylet, then gets into the gut, moves through the whole gut system, and that up into the salivary glands. And it's from there that the aphid can then transmit to another plant. So that occurs over a period of days. And then when it does spread the virus to another plant, it probably needs to feed for maybe at least 30 minutes. But the thing there is that once it has this charge of virus, it's got it for life. And so as it moves through a crop or moves between crops, it still has this virus in its system and can keep transmitting, which is totally different to a non-persistent one, which has to keep getting a new charge of virus as it moves through. So another group of the virus is spread by whitefly. The bigamoviruses are an enormously important group of viruses worldwide, particularly through Southeast Asia. And many bigamoviruses have impact on many vegetable crops, including capsicums. All of them are spread by the silverleaf whitefly, which is what we probably know as Bemisia tabacei biotype B. It's also now called Bemisia argentifolia. But all bigamoviruses are spread by the silverleaf whitefly. It's persistently transmitted, which I've mentioned before. The only real bigamovirus of importance in Australia is tomato yellow leaf curl virus in tomatoes in particularly Queensland. and that entered Australia back in 2006. But the fact that it's here indicates that there is a pathway, and so PMMoV is certainly a biosecurity threat. The good thing about it is that they aren't seed transmitted, and they're probably more likely to come into Australia maybe on produce that's brought in, or for some plants that may be brought in illegally. But certainly a group of viruses which we're extremely fortunate not to have many representatives of, because they're incredibly damaging. And then the next group is viruses spread by contact. And again, we move from insects doing the job to actually viruses that occur at a very, very high concentration in plants. And they're very stable, can reach extremely high concentrations, and they don't require an insect vector for transmission, but they're very easily spread by contact, by contaminated equipment and farm operations. So if you have a capsicum plant that's got tomato or tobacco mosaic or pepper mild mottle virus, if you just brush against the leaves, or if you happen to be cutting the plant to remove fruit, there's huge concentrations of virus that get on your hands and on cutting implements, and you can transmit so easily as you move up and down the row. The other important thing is that they're one of the few groups of viruses that can survive outside of living plants, in dry crop debris, in soil, and in inert surfaces, for example, equipment, the floor of greenhouses, irrigation equipment. They've got this incredible capacity outside of the plant to actually survive, whereas almost all other plant viruses, once the plant dies, the virus dies with it. PMMoviruses are frequently carried on or in seed, and this is very often the source of primary infection in a crop where some contaminated seed is planted, and then because they're so easily spread by contact, they can be moved through a crop very rapidly. So it's some symptoms of PMMovirus on capsicum in southern Australia. Symptoms on the leaves aren't particularly strong, but if you look at the fruit, they're distorted and quite mottled and can't be sold. And of course, if you touch that fruit and then move to something else, you'll spread the virus extremely easily. So management of virus diseases in capsicum, what are our options? Well, certainly one is having a good farm biosecurity plan. I think looking at the ways in which these viruses can be spread, we certainly need to be careful about visitors, staff, transplants, seed, and equipment that comes onto the property, to make sure that we're not allowing some of these pathogens to be brought in on that material. On farm, manage the weeds, which are hosts of both the insects and the viruses. And if you can do this across a group of farms or a district, that's even better, because most insect vectors are mobile. They're not going to hang around just on your farm. They'll move through an area if they can. So if you reduce their opportunity to breed and to maintain themselves, well, that'll make a huge difference. Excuse me. Variety selection is important, because there are capsicum or commercial capsicum varieties which have good resistance to most of our viruses infecting capsicum. There's ones with resistance to tomato spotted wilt, PVY, cucumber mosaic virus, and the PMMoviruses. Unfortunately, there's no resistance commercially available for capsicum chlorosis virus. And obviously with variety selection, there's more than just disease resistance involved. But if you have an issue with a particular virus, there's every chance that a few of the varieties will have resistance, which is very useful to you. With the tomato spotted wilt resistance, it is fairly vulnerable to breakdown, but in most cases in field production in Australia, it's still effective. And we can help maintain that, of course, by reducing the amount of virus pressure that it's under. So less pressure, then there's every chance the resistance gene will last for longer. We need to monitor insects and also find out what virus we actually have in the crop. So if you know what insects are around, what virus you have, then you can decide sensibly on what controls you really need. That may involve insecticides, or in other cases, there'll be of very little use because they either won't work quickly enough, or it could be a TSWV virus which is spread by contact. So just to close the formal part, I'd certainly like to acknowledge the Horticulture Innovation Fund and people in each state who've been on the area-wide management project now for 4 years, and it's been a large team that's done a lot of work, and we certainly appreciate everybody's contribution. So thank you.
Excellent, thank you, Denis, and some great insights there too, particularly on the insect transmission as it relates to this particular crop, which has been really interesting, and hopefully you've taken away some useful tips and tools. We're now going to shift for the remainder of the session to Q&A, and that's a chance for you to ask questions of Denis, either specifically about the content that's been presented today, or it might have been something on the wish list that you were thinking about in the lead up to today's session. We've already had a couple of questions come through. The first one is from Michael, and Denis, Michael's just asking, what is the mechanism by which TSWV viruses can last so long on non-plant surfaces? Are they not affected by temperature or UV?
Yeah, the TSWV viruses, that really is interesting. It's the fact that they occur in such enormous quantities, even in a crystalline form, that UV, for example, will affect them. I think the concentration is such that they can still resist that. They do best probably on dry debris that may be protected to some extent in a greenhouse or in an area which doesn't get as much UV. They still seem to be able to survive on cutting tools and implements, even in the open, for quite a number of months. So they do have this level of protection against being disintegrating in the presence of UV and other chemicals. They don't do as well if they're in soil which is kept pretty wet, because I think microbial degradation seems to affect them a fair bit. But yeah, it's incredible that this one group of viruses is basically so tough and can survive so long in a very, very harsh environment.
Which just highlights the importance of that contact spread that you mentioned before, Denis, and things like farm hygiene as well. Another question has come through as well, Denis, just around identification of symptoms and how easy that is to do in the field to determine which virus you've actually got.
Yeah, it's pretty difficult actually, because I think if you look at the symptoms of viruses in not only capsicum but many crops, they're really a combination of mosaics and mottling and stunting, and in quite a few instances you can have more than one virus within a crop. So I think if you've got a virus problem and you really need to think about how you're going to manage it, the first step is to find out which virus you actually have, and we can do that pretty easily through the testing services which are offered in each state. There are also the little dipstick tests which you can do, but I'd think that if you have a real issue, well, then get some representative samples tested by a good virology laboratory who can tell you which virus is there, and then you can start basing your management around that knowledge. Sometimes we have problems working into a farm and just saying, well, this is virus X or virus Y. So it really is difficult to separate out all those individual symptoms.
Thank you for that. Our next question comes from Cameron. Thanks for sending that through. Just wondering, Denis, whether you've done any work on plant biostimulant products that may boost the plant's ability to defend virus infection or at least reduce the severity of the virus?
No, we haven't. Certainly, I think the thing with virus infection is that the infections are systemic, so it will spread through all of the plant. Some biostimulants or fertilizer can certainly have two effects, I guess. They can, because the plant grows very rapidly, it may produce more produce for you. On the other hand, you can find that a very rapidly growing, very healthy looking plant is actually also a great host for virus because it's able to multiply and spread systemically very rapidly. So I don't really hold out a lot of hope for biostimulants as a really good management technique for viruses.
Thank you for that. Another question we've had in is just around the difference between protected cropping environments versus open field and some of the risk factors. Denis, I was just wondering if you could talk to those two production systems and perhaps some of the key things growers might need to look out for, particularly in protected cropping systems.
Yeah, well, certainly in protected cropping, that's where we have more outbreaks of the TSWV viruses than in field situations. As I've mentioned, they're so easily spread by contact that in a protected cropping situation with a lot of plants together with many, maybe several different greenhouses, it's so easy if a TSWV virus is introduced for it to be moved up and down the row during harvesting and growing operations and then moved from house to house on equipment and clothes. So yes, certainly protected cropping and TSWV viruses often go together. In field situations, I guess it's the insect carried ones like cucumber mosaic and the TSWV viruses like tomato spotted wilt virus, which are more likely to be a problem where you have thrips and aphids able to move over quite a wide area. That said, if you do get tomato spotted wilt into protected cropping, and that does occur, if you return capsicum on capsicum, well then often the thrips remain in that greenhouse during that lull period and are ready to infect the young seedlings as they come through.
Yeah, so highlighting the importance then too of things like crop rotations and just management there as well, which is a bit more difficult, isn't it, in the protected cropping, more intensive systems?
Yeah. Yep.
Denis, you mentioned during your presentation just the control mechanism of insecticides. I was just wondering if you could go into a bit more detail about deciding perhaps which insecticides to use or whether to spray or not, based on whether it's persistent or non-persistent, and just how effective some of those will be.
That's right. Well, certainly, and this is where the type of transmission is really important. With non-persistent borne viruses, applying insecticides for aphid control is usually not very useful because that spread process is just so rapid. Certainly if you've got really large aphid populations on the crop, some strategic spraying to reduce that's important, but in many instances with non-persistent transmission, it's aphids that are moving quite quickly through a crop and very briefly feeding. They're actually getting the virus and then moving it rapidly as they move through a crop. With things like tomato spotted wilt, we have persistent transmission. Certainly, one of the problems with spotted wilt is that quite often infection is incoming, that you get a flight of thrips, which brings it into a crop, infects a lot of plants at one time, and then that generation goes or declines. If those thrips remain in the crop and continue to breed, well, the insecticide will certainly reduce the amount of virus transmission because it'll affect the next generation of thrips. So I think with persistent viruses, you've got opportunities with insecticides to to actually reduce spread effectively. With the non-persistent viruses, it's really difficult with an insecticide to do a lot because the transmission process is just so rapid, and very often it's the aphids that are moving in and out of the crop really quickly, which are doing most of the harm.
That's really great. Thanks, Denis. And thank you everyone for joining us today too, and for those questions. Half an hour really does go quickly and we are at time. But the key takeaways, I think, from that is the importance of monitoring to manage and knowing what you do have so you can manage it correctly. And Denis mentioned the importance of getting samples tested so you can pinpoint exactly what you're dealing with. Also some really good practical tips and tools in there around farm biosecurity and hygiene management to prevent the incursion and spread of those particular viruses. As I mentioned at the start of the session, there are some handouts on the control panel there for you to download and take away today if you haven't done so already. Feel free to jump onto the Soil Wealth website at soilwealth.com.au for any additional resources and to sign up for the next webinar in this AWM mini-series, number 3 in October on lettuce viruses. I've been Carl Larson. A big thank you to Denis Persley from QDAF for joining us and to you, and we look forward to seeing you at the next session. Thank you, everybody. Bye-bye.
Yeah, thank you.